My name is Lilly, and I am a scientist up at OHSU, and I am also a member of the Monarch Initiative. And I will be telling you a little bit more about what Monarch is and how we are dippering the data, open source tooling for data integration. And at Monarch, we are integrating data from diverse species and sources with the main goal of disease discovery and diagnosis to try to improve patient health. So I'm going to start with a little outline. I'll tell you who we are at Monarch, what we're doing and why, and then I'm going to define some terms that we might be unfamiliar with, such as phenotype and ontologies, and then I'll get into our extract, transform, load workflow, and then I'll end on some data tool examples to show you guys how we're using this data that we've integrated. So Monarch is a large international consortium. This is just an overview of some of our collaborators and stakeholders. We're really involved in open data and open science and passionate about making our tools available for others to look at. I encourage you to go to our website and also our GitHub page if you want to learn more and see our code. So this is just an overview again, and Monarch is trying to be fully translational, which means that we are taking basic data produced by researchers and trying to translate it into the clinical spectrum to affect human health. The big problem that Monarch is trying to solve is that there's a lot of data that's being produced that isn't being shared, and it's being underutilized by both researchers and clinicians. So at Monarch, we're trying to make all the data count and really integrate it so that we can use it. And one of our big goals is to integrate data, form a database, and use this database to support disease diagnosis, especially for rare diseases. And I say rare diseases, and you think it might be a small problem, but rare diseases actually affect a lot of people. On average, 1 in 10 Americans has a rare disease, which comes out to about 30 million people in America alone, and about 7,000 different diseases. And importantly, only about 5% of these diseases have a known treatment. And they're also really hard to diagnose, and one of the reasons that they're hard to diagnose is because they're rare. You know, a patient comes to a doctor and they have a set of symptoms that that doctor may have never seen before, and so the doctor doesn't know how to diagnose that patient because they don't have any comparison points. So at Monarch, we're trying to integrate data that we can give to clinicians, for example, to add those comparison points back in. All right, so one of the problems that we encounter when we're trying to integrate the data is that data sources are diverse, they're distributed, and they're differently modeled. I used to do neuroscience research with flies, and so our data is very different from that data that clinicians produce. You can imagine a scenario where a clinician has a Parkinson's patient and they have data related to that human, and then there could be researchers doing mice or fly data or even chicken research in Parkinson's disease, and they might have some breakthrough that isn't being translated into human research because it's really hard to integrate that data. So besides these problems, we also have data that is in physical and semantic silos. As you can imagine, like I mentioned, you have different research groups that are all producing this different data, and they don't share very well. Frequently, data is only shared when it's published, and then only the positive results are shared, and this can take a really long time to write a paper and get it published for anyone that's been through that cycle. But there are databases that have published data and sometimes unpublished data that researchers and clinicians can access. But even then, the data is still semantically siloed. And an example of what this would be is, say, like a lot of anatomy terms, like in humans, we have data about the hand, and in mice, we have data about a paw. And we know that those two things are synonymous, but a computer doesn't innately know that, so we can't integrate over that without telling the computer that those two things are the same. Here's another example of what a semantic bridging challenge looks like. So say we have a model organism researcher who is studying frogs, and he has a frog that has a short jaw, bulging eyes, trouble breathing, and spider fingers. And say this researcher knows the genetic cause of all of these symptoms, And then we also have a patient that goes to the doctor and has the exact same symptoms. They match each other. But the doctor or the geneticist does not know what is causing these symptoms in the patient. If we're able to integrate this data, then we might be able to figure out, look and see if that genetic cause in the frog is similar to what's causing it in the human. So this is just a very, very simple example of what we're trying to do. So each of these terms here that are in the speech bubbles, I've been calling them symptoms, but in the biology medical world, we call them phenotypes. And phenotypes are the outward presentation of our genetic background combining with our environments. And so to help you guys understand more what I'm talking about when I say phenotypes, I have a picture. So this is showing us different phenotypes. Here you have blue skin, or really big ears and you're bald, or striking facial features. So all of us, if you look at each other, we all have different phenotypes. And when someone has a disease, they present at the doctor with a collection of different phenotypic features or symptoms. So I'm going to talk about phenotypes, and I just want you guys to remember what that term means. And for one other example, I'm using the unofficial theme of CSV, which is cats. This is Maru, and Maru is a famous internet cat. Maru loves to jump in boxes. And so we could describe that behavior as a phenotype of abnormal sheltering behavior. So if Maru was a patient, this is the kind of data that we would collect on Maru. We have his biography. He's 4 years old. He's a male cat of the Scottish Fold breed. We also have some measurement information about him. He weighs 6 kilograms. And then our phenotype and qualifiers is that the abnormal sheltering behavior, the onset of birth. And then, of course, very importantly, we have our source. In this case, is from a YouTube video, which isn't normally what we have. So this gives you an example of what the kind of data that is collected from patients would look like, and some of the type of data that we are trying to integrate at Monarch. Another reason why I'm showing you Maru, besides that it's a cute cat and that's kind of the conference theme, is that it's really important to integrate data from all species, because adding other species data helps us fill in knowledge gaps in the human genome. So for example, I was a neuroscientist, and a lot of basic neuroscience research was first done in the squid. So it's really important. We can learn a lot from integrating data from other species. And another reason why it's really important to integrate data from other species is because other animals can have the same rare diseases that humans have. Here's an example of a little boy with vitiligo, which is a rare skin disorder, and also a dog that has the exact same rare disorder. And these two found each other because the little boy's mom was searching for his phenotypes or his symptoms online. She was Googling and ran across the same phenotypes and symptoms in the dog. And so this is just an example showing you how we can find different data sources from very diverse areas that you wouldn't normally think of. But as you can imagine, the phenotypes that are used to describe the dog are different from those used to describe humans. So this is part of the semantic challenge of integrating this data. So to integrate terms that are semantically different, we use ontologies. And there was a really great talk yesterday that involved the swantology. I don't know if you guys heard about that. But my colleagues made another slide to show you guys what an ontology is. An ontology can be described as a knowledge classification comprised of terms that are formally defined and logically related. Or it can be thought of as a dictionary of terms and how they're related to each other. Ontologies are used for a variety of things, such as Amazon uses them, and the actual Amazon uses them too. But today I'm going to tell you about a beer ontology, because we're in Portland. So we have different terms here, like light beer is a type of beer, so the is a is showing us a relationship. Oops. And then we have Pilsner is a light beer, Lager is a light beer, Porter is a dark beer, Stout is a dark beer. So this is a simple example, but it's showing you that ontologies semantically map out the relationship between different terms. And they're used to take really complex structures and break them out into different patterns. We use ontologies to integrate our data. And at Monarch, semantics serve as a bridge, with ontologies acting as our universal converter box. So we're able to use this universal converter box of ontologies to integrate data and map relationships between genes, phenotypes, and diseases from all species, including humans. So how do we integrate all of this disparate data? Well, we use our extract, transform, load workflow. Our workflow is called Dipper, and this was developed by my colleagues, and you can read more about it at our GitHub site if you'd like to. Dipper stands for Data Ingest Pipeline. It is an open source Python package, and Dipper extracts various types of biological data like genotype information, phenotypes, and even evidence information, such as a paper that was published that we got the data from. Dipper then transforms that data into RDF triples, which stands for Resource Description Framework. At the core of RDF is a set of triples, or subject, predicate, object, which is kind of like the ontology I showed you earlier, where the predicate is a relationship between different terms. And the subject, predicate, object in an RDF triple makes up an RDF graph. So once Dipper transforms the data, we get data that is graph-based, queryable, Sparkle, and it's also human readable. And then finally, we load this data into our BioLink API and then use the data for various tools. I'll tell you a little bit more about this now. So first, we extract data from multiple sources. We have very diverse data, information about genes, mechanism and context, and phenotypes and diseases. And then we take this data from very disparate sources, such as genomic resources, model organism databases, which are where researchers can store some of their data, and also from clinical data, and then from published papers. So as I said, I used to be a neuroscience researcher, and I studied the fly. So I'm going to show you an example of what this data looks like from the fly model organism database and also the mouse model organism database. And I'm going to show you guys the gene that I used to study, which is called falafel. So in flies here, you can see at the top there's the symbol and then the name. And this information is also found on the mouse resource. And this is just a screenshot showing you a small amount of the information that this contains. There's a lot of information here. And I've tried to highlight the commonalities between these two sources with red arrows. So you can see that there is some similarity, but there's also a lot of difference. And especially if you look at the symbols, you'll notice that they're different between the two species. So like in flies, the gene I studied is called falafel, but in mice, it's called PP4R3A. And I know that they're the same gene because I studied them. But once again, the computer doesn't know that these are the two things. But we use our Dipper code to tell the computer that they are actually the same, and we use ontologies to bridge that semantic gap. The really big arrow that I've drawn over here is showing us the common NCBI gene number, and this gene number is the same no matter what species you're in, and that's what we are able to integrate this data on. So for each type of data, we find one commonality and integrate across it. All right, so once we have found the data that we want to extract, the next step in our pipeline is that we semantically map out this data. And this is what this looks like. It's an example of looking at genetic data. And this is the RDF triple again with the subject, predicate, object, where the subject here at the top is the gene, and then the predicate is the relationship. So I wish I was a little taller. If we look at that one, the relationship there is subsequence of, and then chromosome is the object. So we're saying that a gene is a subsequent of a chromosome, and a chromosome, for instance, is a member of a reference genome. So this is how we're semantically mapping out the data in this pipeline. And I want to mention that these terms here come from various ontologies. For instance, you see the RO with the colon, that's the relationship ontology. All right, so we use various ontologies to transform the data. Down here in the colored area, we use Geno for genetics, Uberon for anatomy terms, such as hand and paw. We use Go for gene functions, Uberpheno for phenotypes, and Mondo for disease terms. And we use a few other ontologies as well, but these are the main ones. And I want to bring us back to that NCBI gene number that I was talking about a minute ago that we're comparing upon. So say we could use Go and that gene number to figure out the relationship between the gene that I studied and any cellular processes. All right, so once we have extracted and transformed our data, we load it into our BioLink API. And you can check out our API at both of these links here, and then here's our GitHub for the API. And this is just a screenshot of what part of our API website looks like. And there are several types of queries that we can do on this page here. For instance, say you want to figure out, you want to query for human and mice data that have abnormal eyelid phenotypes. Or if you are a worm researcher, you could look for all worm models of Parkinson's disease. So those are just two examples of the types of queries we can do with this information. Also, if you're interested in a particular disease, you can go to our website and type in that disease and find all of the data that we have integrated about that disease. This is an example of the data that we have on Marfan disorder. And you can get to this by going to our website, or you can go to this link, or you can go to our website and just search for Marfan disorder, or search for, say, a different disease or a phenotype. And this is an overview page showing us, again, these RDF triples. So we have the subject, relationship, object. And then we also have information like what species it came from, any evidence related to that data, the reference, and then the database source. And then we can click on each of these gray tabs at the top to see that for our data, we have 375 phenotypes that are associated with Marfan disease. We also have genetic information, genotypes, other model organisms, variants like genetic mutations that are all associated with Marfan disorder. Finally, we can click on that compare tab there on the end, and that brings up our PhenoGrid tool that allows us to compare phenotypes between Marfan disorder and other disorders from humans and other model organism data. All right, so my final example of one of the tools that we use with our data is called Exomizer. And Exomizer is a tool that has been collaboratively developed by us, the Sanger Institute, and Charité. And Sanger uses genetic information to try to find causes of disease in humans. Importantly, it doesn't only use genetic information, it integrates the genetic information with phenotypes from humans and model organisms. And previously, Exomizer has been used to diagnose 2 patients that were undiagnosed by traditional methods. Like they went to the doctor and the doctor couldn't diagnose them. And currently, the 100,000 Genomes Project in England is using Exomizer to try to diagnose lots of rare disease patients. So the takeaway from this is that when we integrate genetic information with phenotype or symptom information from humans and model organisms, we're able to have a real impact on human health. So with that, I want to thank all of my colleagues. As I said, we're international, so we have colleagues from around the world, and we're always looking for feedback. Part of my job is to collect feedback and help users with our tools. So please go play around on our website, and if you have any feedback, you can email me. I'm winfree@ohsu.edu. You can also find me on Twitter, and I'm running the Monarch Initiative Twitter account too. Also, I'm going to put in a little plug that we are always looking for new people to join our team. So if you're interested in developing tools like this, just contact us. You can contact me, or you can email our info address and let us know. And we're always looking for new talent. All right, I'll take any questions. Nope, go back.
I'm curious what are the challenges you're facing when you're continuing to next, where's like the edge of the tool or the things that you're trying to do?
There are several challenges. Yeah. Like where to start? So as you can imagine, the data is very diverse, and there are so many different sources that we're pulling it from. And so, like I mentioned, let's find this. So every time that we pull in a source, my colleague Matt maps out this data, and this can be time consuming, and so that's definitely our like next step of improvement, is figuring out a way to take out this step kind of and integrate it better with our main coding people. So that's part of it. And then we're also always trying to improve our quality control and quality checks after we've integrated the data, to make sure that we aren't making, we aren't adding in a relationship that isn't true. And then, yeah, we're working on new tools. We have a tool where you can put in a PDF of a paper, and then it will pull out phenotypes by using natural language processing. Yeah, and so it can pull out positive phenotypes really well right now, and we're working on making it understand like decreases or negatives, like they do not have this phenotype. So that's being developed right now too.
Sorry, I thought you had the fly base up there. Are you working with the learn base or Expresso folks?
Yeah, we are working with Warren Base some. I don't actually know the second one.
Well, I'll follow up with you about that, because you're doing a lot of similar ontology and marking straight up from the PDF.
Mm-hmm.
Identifying words and ontologies in the PDF.
Yeah, let's talk about it later.
They're like kind of the back end of Warren Base.
Yeah, I mean, I bet we are, but yeah. Mm-hmm.
Yeah, just a quick, just to get a sense of time scale, how long has this been in development? How long does it take to get this amazing network of
Yeah. So Monarch's been around for about 5 years. One of our PIs is Melissa Handel up at OHSU, and she's been working with zebrafish database for a long time. Like Robin probably knows better, but at least 5 years under the Monarch name.
So you really demonstrated to us the impact that this can have when people share their data, and it's also better when you connect up and you can actually diagnose people better. Do you ever present this to researchers creating that basic research data? And if so, does it incentivize them to think more carefully about how they're communicating that data and that research?
That's a really good question. I would like that answer to be yes, but I don't know that it necessarily does have that much of an impact at the data creation level. And a lot of the data that we're importing is from model organism databases, so they've already taken the data and kind of converted it into their style, but hopefully it would affect that. Yeah. One more question, or should we go? Okay, thanks you guys.